Okay, so we are going to get into nerve cells now, and we first need to talk a little bit about the discovery and the ability to um, look at nerve cells with microscopy. So here is Golgi, and he was an Italian physiologist who figured out basically how to look at nerve cells by staining them. Oh, where's my laser pointer at? There it is. What the heck? Okay. So Golgi figured out a way to stain nerve cells using silver. And these are new pictures. Like, this isn't microscopy from Golgi's time, but nonetheless, these are neurons that are stained with what's called a Golgi stain. And the Golgi stain uses silver to stain, as the name suggests, the neurons into this opaque color. So you can then use a microscope and get really good resolution, assuming that your microscope has the required level of zoom on it. But we can see here, we're going to go over the different parts of a neuron, but we can see here the soma, and then we can see dendrites, and then zoom in on some dendrites, and then a huge crazy zoom on some other dendrites here, and you can actually even see dendritic spines. So dendritic spines are these little outgrowths on the dendrites themselves that increase the surface area of the dendrites to receive communication from other neurons. So Golgi is a really famous guy in neuroscience, very important. And Ramon y Cajal is often thought of as the father of neuroscience, or maybe at this point the grandfather of neuroscience. He was a Spanish neuroscientist and anatomist, and he discovered that cells of the nervous system are not contiguous, which means that they're not actually touching. So he used the Golgi stain, and he wanted to be an artist when he was young, and his dad said, "Bro, that's like kind of not very smart. You're not going to make any money. So go be a doctor." And so what he did was he became a scientist and a neuroanatomist. So these are actually his drawings over here. And so you can see that they look pretty similar to these microscopy images, honestly, which is pretty wild. So here's, again, the microscopy images from like a Golgi stain today. Here's what Ramon y Cajal was drawing back in the day. And so what he discovered from his observations is that the nerves of the nervous system are not touching. So back in the day, there was kind of a feud between Golgi and Cajal, because Golgi thought that the neurons were touching and that all of the information that was being spread was through electrical communication that was driven by direct physical touch between the nerves. Ramon y Cajal, based on his microscopy data and the fact that when he was looking at these cells, he felt like they weren't actually touching. He thought that he saw a small gap in between the two cells, which he did. He and Golgi actually got into a bit of a scientific spat when they were winning their Nobel Prize. And they basically used their Nobel Prize speeches to kind of like trash the other person, and be like, nah, he's confused or whatever. But in any case, it would turn out that Ramon y Cajal was correct, and Golgi, who I am obscuring the face of, was wrong. Neurons actually, for the vast majority of them, are not touching. There is a small gap between the two cells, and this gap is referred to as the synapse, which is where the communication between neurons is happening. Okay, so here's a neuron, and we're going to start getting into some cell physiology, and I don't want to freak anybody out, you know what I mean? Like, I know that there's going to be a lot of information here, but I'm not going to be asking super in-depth questions about this kind of stuff. Aside from the cell membrane, the neuronal membrane, which we're going to have a whole lecture on in of itself, is going to get pretty complicated and in-depth because it's very necessary to how the cell functions. But for the rest of them, honestly, you're going to just have a very shallow understanding of, because this isn't a molecular cell biology class. It is biological psychology, so we're not going to go super in depth. So you only need to know what I'm basically, the little bit that I'm going to talk about. I'm going to touch on cell membranes right now, and then the next lecture, I believe, is going to be much more, whatever lecture it is, I'm pretty sure it's the next lecture, but when we start talking about action potentials, it's going to be much more in depth than what we're talking about now with the cell membrane. But I'm just going to say that there's going to be a lot of jargon and scientific terminology being used, but try not to zone out, because this is going to be the hardest section is the cell membrane section. Right now, talking about the organelles, we're not going to go super in depth, but anyway, this cell membrane, this is a kind of a representative diagram that you'll see, forever, which, mine I just drew, so it's not very good looking, but you have essentially a zoomed in neuron, and we have the cell membrane here, being represented by this black line, and then the extracellular space is just referring to everything that's outside of the cell. And then we have the intracellular space, which is referring to everything that's on the inside of the cell membrane, right? So the cell membrane itself is composed of a phospholipid bilayer. So it's called a phospholipid bilayer because there's two of them, right? So there's two layers, bilayer, and it's composed of phospholipids. So a phospholipid is this particular chemical compound, and they have chemical properties. And that property that is relevant to you is that the heads of the phospholipids are hydrophilic, meaning that they are attracted to water. And the tails, these little yellow legs, are hydrophobic, meaning that they repel water. And it's actually the fact that these chemicals have these two components that form the cytoskeletal structure of cells, generally speaking, but also neurons. So the reason why cells are circular is because of the fact that we have this phospholipid bilayer that composes cellular membranes that repels water on the heads and attracts water on the tails. And that makes the cells kind of form this circular cytoskeletal structure. So we're going to go way more in depth about this again in the next lecture, but for now we will just try to really, you have to know this. Cellular membranes are composed of phospholipid bilayers. Phospholipids are a particular type of chemical made of phosphates and lipids, and the components of this that's relevant is that they have a hydrophobic tail and a hydrophilic head. Okay, so you must know what hydrophobic means. You must know what hydrophilic means. You must know that the cellular membrane is composed of a phospholipid bilayer. Okay, so neurons have a lot of unique characteristics. As you can see, this neuron is a very peculiar looking cell compared to like other animal and eukaryotic cells. Or eukaryotic organisms that have animal cells. So we need to know here, though, that neurons, although they have a lot of unique characteristics, they also have the same organelles of other animal cells. So they have a nucleus, okay? And the nucleus, what you need to know about the nucleus, there's a lot of like things that are important about nuclear signaling and stuff like that we're not going to get into. But for your purposes, you must know that it's a large organelle that contains the DNA. So the instructions for production of proteins, so we have here produces precursors for protein synthesis. The information that is going to produce more proteins and function is in the DNA, and that DNA is found in the nucleus. So you must know that neurons do have a nucleus, and in the nucleus, that's where we find DNA. You must know that there's mitochondria. Mitochondria is a thing. It's the powerhouse of the cell. We can see that right here. And although I'm showing you just a typical animal cell, I'm just showing you that because the organelles of an animal cell and a neuron are similar. So mitochondria produces adenosine triphosphate. So again, a lot of these terms, and I'm just explaining this, some of you might be like, dude, this is stupid. I remember this from, like freshman biology or chemistry or whatever. But like for me, I didn't know any of this when I first took biological psychology because I was a psych major and I had no introduction to science at that time. So, or any of the like traditional STEM fields. But in any case, adenosine triphosphate is called that because it's made of 3 phosphates that are attached to an adenosine molecule. So adenosine is a chemical, phosphates are a chemical, and there is 3 phosphates attached to this adenosine molecule. So mitochondria produces ATP, adenosine triphosphate, and that is how the cell is using or producing energy, is by removing one of those phosphate groups. So you'll also see at some point adenosine diphosphate. It's just saying that, we took this adenosine triphosphate molecule, took off one of the phosphate groups, and in taking off that phosphate group, we produced energy. And that's the heat or the energy or whatever that was used. Okay, so we have mitochondria, and mitochondria produces ATP. ATP is an adenosine molecule with 3 phosphate groups attached to it. So this just means that in order for the cell to function, in order for the cell to have energy, it needs the ability to have this molecule to remove phosphate groups to produce that energy. So you need to know that the mitochondria produces adenosine triphosphate, and this is how the cell produces energy. Ribosomes, which are these little dots that are basically all over the place, translate messenger RNA into chains of amino acids, which ultimately fold and function as proteins. So if you don't have basic chemistry, then you should just know that what proteins are made of. Proteins are just chemicals that are made from amino acids, and they have particular components, So it's like if you have a chain of amino acids, that doesn't make it a protein. But once you get to a certain length of amino acid, and then you have a couple of things that are on the ends of those chains, they will fold together, and then that is what a protein is. And depending on the characteristics of that protein, it will have certain functions. So what you need to know for this exam, though, would be that ribosomes translates messenger RNA into chains of amino acids, which ultimately fold and function as proteins. There's also the endoplasmic reticulum, which is this thing right over here. And it's important for several things. Protein synthesis. Again, protein synthesis is just taking the DNA instructions and then producing, again, these long chains of amino acids that fold in on themselves and then eventually have a function. So that's protein synthesis. And then there's lipid metabolism. But what's really important for us as neuroscientists is the intracellular calcium. So I'm going to be asking specifically about where intracellular calcium is stored, and the answer to that would be in the endoplasmic reticulum. So you must know that the endoplasmic reticulum is an organelle of all animal cells, and it stores intracellular calcium, and that's really important because, as you'll find out later when we get to synapses and things like that, calcium is super important for neuronal functioning. Okay, so let's go ahead and take a look at the structure and function of neurons. This is a typical looking neuron. It's just what canonically, if you're going to draw a neuron, it's going to look something like this. And there are many things that are unique to neurons that you won't find in other animal cells, such as dendrites. Dendrites are these outgrowths of the soma that receive information from other neurons. So the more dendritic arborizations that they have, the more surface level there is for other neurons to communicate with this cell here. And we can see that I put this little neuron here to just illustrate canonical connections between cells. So there's a lot of different ways that neurons actually can hook up to each other. It can be from axons to dendrites. It can be from dendrites to dendrites. I mean, there's all sorts of different things. Or different ways, but like the canonical traditional way of thinking about this is that the dendrites of one neuron are communicating with the axons or the axon terminals of a previous neuron. So we have dendrites, and dendrites, like a lot of names in science has to do with like Latin. So, oh man, my face is covering the, oops. There you go. Now you can see the title in all of its glory. But in any case, a lot of names and vocabulary in science has to do with like Latin origins. So dendrites has something to do with trees. You know what I mean? Like dend is like a prefix that has to do with trees. So they think that the people that discovered these things kind of felt like they looked like arborizations, like trees. And, they kind of do, so they call them dendrites. We also have the soma, which again is going to be the same as what you would expect in a normal animal cell, which is the body of the cell. Soma means body, so it's the main body of the neuron. Within the soma, that's where we have the nucleus and also all those other organelles that we previously discussed. The mitochondria, the ribosomes, the endoplasmic reticulum, etc., along with the nucleus is all there in the soma. Now, neurons and neuronal communication is very unique. It has a lot to do with electrical signaling. And as we're going to get into later on when we get to action potentials, we'll talk potentially a little bit more about that. But we have the connection between the soma and the axon, which is this thing that I'm about to explain, is called the axon hillock. So you need to know, there's things on this slide that I'm talking about that are not written. So I'm just like putting this aside here. So what is it that you need to know for the exam? You need to know, like, if I said that dendrites, for instance, are there for to receive information from a previous neuron, you need to know that. Like, that's very important. So it may not be, I'm saying this because it may not be obvious to some people, because I'm talking a lot. So it's like, oh, well, what is it that I need to know? You need to know what the function of these things are. So we have dendrites. Dendrites receive information from presynaptic cells, right, or other neurons. The dendrites are attached to the soma, which is the body of the neuron, which contains all the organelles, including the nucleus. And then we have a point where the soma meets the axon. That's called the axon hillock. And there's an important function of the axon hillock, which is that it consolidates all of the information that the neuron is receiving, and it decides, obviously it's not conscious, but there's going to be some decision here, for lack of a better word, that will say, okay, we're going to send all of this information or not send all of this information down the rest of the axon, and we'll get into what that information is during the action potential section. So you need to know that the axon hillock is where the soma meets the axon, and you need to know that this is the integration zone. The axon hillock is the point at where the soma is going to basically send the electrical information or not send the electrical information down the axon. So we also have the axon, which is this weird looking tail thing that has fingers on the end of it. And the axon is going to be surrounded by myelin sheath. Myelin sheath are these like fatty deposits that are fatty membrane. It's made of fat, literally, and it's wrapping around the axon. And the reason why it's here is because the axon communicates electrically to the rest of the neuron. So in order to increase the speed of that electrical signal, this myelin increases conductance. Okay, conductance is the ability of the cell, or the speed at which the cell, or the particular characteristic of the cell that makes it viable for electrical transmission. Okay, so myelin wraps the axon, and it increases conductance for electricity. Very important. I mean, obviously, that's for sure going to be a question. Now, in between the myelin is something called nodes of Ranvier. So the nodes of Ranvier are actually these little spaces in between the myelin sheath. So in between the myelin sheath, we have sodium channels. Now, I'm not going to ask you specifically what kind of channels are here, just because that's like a little bit too, I think, too granular. But you do need to know that essentially what's happening is that there are an increase or a how do I say it? Like the electrical signal is strengthened at these breaks in myelin because there are channels that allow the influx of cations, which are, in this case going to be sodium. But you just need to know that the nodes of Ranvier, like what is specifically like you'll need to know is that the nodes of Ranvier increase, again, the speed and the strength of the electrical signaling that is taking place throughout the axon. Okay, so at the very end of the axon, we have axon terminals, which are the long, stringy, finger-like things. And at the very tip of the axon terminals, we have terminal buttons, so it's like if this is the actual axon terminals right here, these like little fingers, like this little thing right here, the tip of the finger is the terminal button. Sometimes it's also referred to as terminal baton. I just call them buttons, so whatever. But in any case, these are all the major aspects of all neurons. Now, I say all neurons, but there are going to be some neurons that deviate from this, and they may not have myelination. You know what I mean? So there are some kinds of neurons that we'll go over in the sensation and perception lectures where there's no myelinated axons. So there are always going to be exceptions to the rule in biology. It's like almost never the case that you can always say that, X thing is always like this. Like there's always going to be some kind of exception, but I'm not going to trick you with anything like that. These are the major components of the cells that you need to understand. We have dendrites that receive information from presynaptic neurons. We have the soma that contains the organelles, including the nucleus. The nucleus is what's containing the DNA, of course. And then we have the axon hillock, which is the area where the soma and the axon connect. And there's going to be a decision whether or not to send electrical activity down the rest of the axon. The axon is this long stringy thing that conducts the actual electrical discharge down the rest of the neuron, and that axon is wrapped up with a fatty membrane called myelin. Myelin increases the conductance of that electrical signal. In between the myelin, we have nodes of Ranvier, which have channels that allow the influx of cations, which are positively charged ions, and this increases the strength and the speed of that electrical discharge. At the end of the axon, we have axon terminals, which are these fingery looking things that are going to connect and communicate with other neurons. And at the tip of the axon terminals, we have the terminal buttons, which are going to house neurotransmitter in things called vesicles, but we'll get to that during synapses. So I explained everything slowly, and then I explained everything kind of really quick. So you need to know not just what's on the slide. You need to know what I said, which is the function of each of these components, okay? So, definitely, like, there may be questions about each one of these things. I might ask you, what is the integration zone? The integration zone is the axon hillock. I might say, what part of the neuron communicates with other neurons? I would say, there could be two answers to that, and I would have to specify in the question, but that could either be the dendrites or it could be the axon terminals. I might say, where are neurotransmitters housed? That's in the terminal button. I might ask you, what increases the ability of neurons to receive information? That would be a little bit of a harder question, but it would be the increase in density of dendrites, because the more or the bigger or the longer, or like we said previously with those little microscopy images, the more dendritic spines you have, the more surface area, which increases the ability of this neuron here to be communicated with. So yeah, there's not a lot of writing on this slide, but this slide has a lot of information that you do need to know about. So there are many different types of neurons, but for our purposes right now, while we're talking about the basic way that your body is interfacing with the physical world, is going to be 3 types of neurons. Sensory neurons, which are communicating or receiving information from your sense organs, right? So we have, this skin receptor, which is going to detect all sorts of different things. Like you have cells, that are going to detect stretch of the skin, that detect tissue damage. There are ones that detect thermo insults, so things that are too hot or too cold, and it is going to send that information back to the nervous system. So you have sensory neurons that are innervating your sense organs, your mouth, your tongue, your eyes, your skin, everything on you that you can feel or think, or you can feel, hear, smell, in your nose, whatever. You have these sensory neurons that are then going to send that sensory information back to the nervous system. So canonically, we have these sensory cells that pick up the information. They then transmit that information to interneurons, which are the intermediary between the sensory neuron and the central nervous system. And then the central nervous system is going to send a signal back, okay? Again, through the interneuron, and it's going to be talking to motor neurons. Okay, now motor neurons are going to be actually innervating the muscle. So we see that this diagram looks very similar to the one that I just showed you, where we have dendrites, we have the soma, the nucleus, the axon hillock, we have the axon, and it's myelinated, it's sending electrical information down to the axon terminals, and then we have the terminal buttons, or here they're calling them the presynaptic terminals, and then those are innervating this muscle fiber and telling the muscle fiber to contract, okay? So if I were to say, what are the 3 main types of neurons, you would say sensory neurons, motor neurons, and interneurons. And the reason why we have that question here is because you need to understand the basics of how your physiology and your nervous system are integrated and are basically, communicating and sending information. I just realized that my head is interrupting the flow of the slide here. Anyway. Yeah, so we have, sensory neurons. I'm sure that was infuriating, by the way, because I'm reporting this thing, but you can't see it. But you can see it now. So there are sensory neurons that are picking up information about the world in your sense organs, right? We have motor neurons, which are telling your muscular system to move as a result of the sensory information that was communicated to your brain through interneurons, okay? So for an exam, you would need to know, if I were to say blank neurons send information to your nervous system, right? Maybe that seems like you don't have enough information to answer that question, but you actually do, because sensory neurons are the ones that are receiving information from the outside world and sending it into your nervous system, right? Into your central nervous system. So I might ask, what are the intermediary neurons between your ability to sense information and your ability to act on that information? The answer to that would be interneurons. If I were to say, what are the neurons that are actually stimulating musculature? Obviously, the answer to that would be motor neurons. So these are the 3 types of neurons. We've talked a lot about neurons so far, and neurons are the main, functional unit of the nervous system. So that's why we spend so much time on neurons. But there's a lot more like nerve cells that are necessary to know about, like glia. Okay, so glia in general are referring to all the cells that are nerve cells that are not neurons. That makes sense. So you have neurons and you have glia, and glia are the support system of the nervous system. So I'll mention this again in like neuroanatomy, in the neuroanatomy section, but just so you know, gray matter is referring to the actual cell bodies that you find. So it's like when you're looking at some particular area of the brain, you'll see a lot of gray matter and you'll see white matter. The gray matter is the densely packed in like somas of the neurons, and those look gray. And then the white matter are the actual tracts, or the the axons that are leaving those somas, and they are white because they have that fatty membrane that wraps the axon. So that's the myelination that's wrapping those axons. So just so you know, gray matter is, if you're ever hearing something that says gray matter, that's referring to the actual nuclei or the somas. The white matter is referring to the myelinated axons. Okay, so what are the glia that we need to know about? We have microglia, we have astrocytes, and then we have oligodendrocytes and Schwann cells. So let's start off with microglia. Microglia are super important, and what you would for sure need to know on an exam, or to say that you've taken an introduction to neuroscience or biological psychology, is you will definitely need to know that they are the resident immune cells of the brain. I mean, like if you're having an immune response or some kind of immunological insult, the microglia are going to be what is mediating that response. So they're the vacuum cleaner of the nervous system, so they're going around and sucking up dying cells and like harmful stuff. Obviously, as I've already mentioned, I'm just going to go through these bullet points, but they're part of the immune cell. They remove viruses and fungal infections. They proliferate after brain damage, so that's actually quite important. If I were to say, how do you know that a microglial cell has been activated? It's because it gets bigger, inflamed, it proliferates, and neuroinflammation is actually kind of a huge problem, and that neuroinflammation is due to these microglia, getting bigger after they're gobbling up all sorts of things that are damaging the brain. They also remove dead or damaged neurons, and they help prune ineffective synapses. So pruning is this idea that you may have these two neurons that are communicating, and they're synapsing, they're having synapses or whatever, and maybe they're not communicating effectively for whatever reason. Pruning means getting rid of those synapses or severing that communication between the two cells. So they help prune ineffective synapses, and it provides negative feedback to neuronal activity also. So that's also important. But what you need to know what's on this slide. You know what I mean? So you definitely need to know that it can prune synapses, it gives negative feedback to neuronal activity, proliferates after brain damage, removes viruses or fungi, definitely is part of the immune system. So that's microglia. It's a super important glial cell. Then we have oligodendrocytes and Schwann cells. So oligodendrocytes are the cells that are actually producing the myelination, and they are wrapping around the axons of neurons. So they build myelin sheaths that surrounds and insulates axons. And remember, the myelin, again, is that fatty membranous tissue that is increasing conductance of nerve cells. They supply the axons with nutrients that are necessary for functioning. And I have two of them. I say oligodendrocytes and Schwann cells. Oligodendrocytes are in the central nervous system. I'm going to say that again. Oligodendrocytes are in the central nervous system. When we go over neuroanatomy, I will explain the difference between the central and the peripheral nervous system. But you need to know for sure that oligodendrocytes are in the central nervous system. Schwann cells are found in the peripheral nervous system, but they have the same function as the oligodendrocytes. Okay, so they're more or less the same type of glial cell. It's just where you find them that defines them. Okay. And then we have astrocytes, which are really some of the most important like nerve cells that we can even, talk about. Astrocytes are super important. So like on the exam, I will ask the most about astrocytes because they are so important. I don't know as much about microglia and oligodendrocytes. I know more about astrocytes, so since that is my bias, that is probably what I would ask more about. It's not going to be anything crazy. I mean, like, you just need to know what I've written here on this slide, but they are star-shaped glia that wrap around dendrites connecting to functionally related axons, okay? So what does that mean? It means that it's wrapping around dendrites of neurons that are communicating. But they also, so they receive blood and oxygen from the vasculature and then provide those nutrients to neurons and glia. So the astrocytes are not only providing nutrients to neurons, but also to other glia cells. And this is the most important thing as far as I'm concerned. I mean, obviously you need everything in order for your nervous system to function, but in my opinion, the most important thing to know about the astrocytes is that they create a tight endothelial barrier. And endothelial is just talking about the type of cell, skin cells that are, or not skin cells, but the type of cells that are composing the vasculature, between the vasculature and the nervous system, called the blood-brain barrier. You absolutely, positively need to know what the blood-brain barrier is. So what is it? It is this tight endothelial barrier that is produced by the feet of astrocytes, and it inhibits the influx of potentially harmful chemicals into the nervous system. And we're going to look at that in greater detail momentarily. But you should also know that astrocytes play a role in recycling neurotransmitters. So you can see that there are these feet that are around the neurons, but what you can't see is that there's also like the astrocyte foot, or the part of the astrocyte is right next to where neurons are communicating, and any of the neurotransmitters that are floating away from that synaptic site get sucked up by astrocytes and then recycled into neurons. So definitely you need to know these 4 bullet points about astrocytes. Okay, so in order to understand the blood-brain barrier, we need to understand a little bit of basic anatomy when it comes to veins and arteries. Okay, so your cardiovascular system is really interesting and quite complex, unfortunately, or fortunately, for test-taking purposes, unfortunately, I guess. But I'm not going to ask you questions that are super in depth about the cardiovascular system, but you do need to know like what is meant when somebody says veins or arteries, okay? Because they are very different things. And generally speaking, when people think about their cardiovascular system and these little tubes that are carrying their blood, they normally are referring to them as veins, okay? But vein has a very specific meaning and is referring to a very specific type of anatomical structure. So we have a diagram of the heart, and the heart is composed of 4 chambers, 2 atria, which are seen here, and 2 ventricles. So let's go ahead and start off with arteries. Arteries carry oxygenated blood to the rest of your system. So to your muscles, to your brain, to all of your vital organs. Arteries are the little tubes that contain blood that is oxygenated. blood, this again, everyone's going to know this, but your blood is what's actually transporting oxygen to all of your organs and your musculature, and the oxygen is obviously a necessary component to all of these things functioning. So you have right here, if we're starting at this heart, you can see that there's this valve here, and it's leaving this left ventricle. The left ventricle is receiving oxygenated blood from the left atrium, and the left atrium is receiving oxygenated blood from your lungs. So I'm not going to ask you about this lung portion, but this is just in case you're interested and you want to know. Essentially, when your veins are returning deoxygenated blood, the blood is actually going through like these little veins in your lungs that are then coming into contact with oxygen, receiving the oxygen, and then it goes back into your heart. So your atrium is pumping oxygenated blood into the left ventricle. The left ventricle then contracts and shoots that oxygenated blood into your arteries. This is the part that you need to know for the exam. So the oxygenated blood is going through the arteries, and as it goes through the arteries, your arteries start to branch off into smaller and smaller components. So first it goes to arterioles, which is a smaller artery. And then from arterioles, which is right here, it goes into capillaries or capillaries. I think it sounds funny when people say capillaries, but in any case, it goes into these capillaries, which are even smaller. I mean, super small little tubes that allow the blood to deliver the oxygen. So why is this relevant to the astrocytes? It's relevant because this is where the blood-brain barrier is having its action, okay? So the axons, here's this blood vessel, right? It's an artery, and it is delivering blood and oxygen to the system, okay? But because of the astrocytes, it is bouncing, because of the blood-brain barrier that's produced by the astrocytes, it's bouncing certain chemicals off of it, okay? It's saying, no, you can't come in. So I explained all this just because you need to know that, one, arteries are what is transporting oxygenated blood, and it's doing it through capillaries. Capillaries are just a smaller division of the arteries, okay? Then this deoxygenated blood is going into the veins, and the veins are going to return that deoxygenated blood to the heart. The heart is going to then pump that into the lungs oxygenate the blood, and then the blood, is then recycled. So again, what you need to know for the exam is you need to know that arteries carry oxygenated blood, you need to know that veins carry deoxygenated blood, and you need to know that capillaries or capillaries, whatever this is the part of the vessel or the vasculature that is going to allow the delivery of the oxygen. Now, the blood-brain barrier is going to be found at these capillaries, and it is formed there by the feet of the astrocytes. So I didn't know that I had this slide here, so let's go ahead and see what it says. Blood flows away from the heart through the arteries. Yeah, absolutely. Arteries then branch into smaller tubes called arterioles, and eventually to capillaries that actually are innervating major organ systems and delivering oxygen and other nutrients. Yeah, so so definitely here's just a little summary slide that I made for you guys just to make it a little bit easier to study, because I tend to be a little bit long-winded when I speak, but it is the capillaries that the blood-brain barrier is created. It's at the capillaries that the blood-brain barrier is created through the astrocytes. Yeah, so it's like the astrocytes are wrapped around these capillaries and not allowing the influx of certain chemicals. So here again is just a picture of a blood vessel and what is essentially happening with the blood-brain barrier. So the blood-brain barrier allows certain chemicals through, Fat-soluble molecules like carbon dioxide and oxygen pass easily, while amino acids and glucose need active transport mechanisms. So active transport mechanisms are talking about proteins that actually that act as like a pathway for these chemicals to flow through. Large molecules and charged molecules are inhibited from passing through the blood-brain barrier. So large molecules are going to be talking about like certain types of drugs, and charged molecules also are going to be certain types of drugs and chemicals. So here's some examples of how the blood-brain barrier is working. Heroin versus morphine. So heroin is a drug that actually, once it's in the brain, it turns into morphine. So both heroin and morphine, because they're basically the same thing, are drugs that work on your opioid receptors. Heroin is more fat soluble though, so it passes through the blood-brain barrier faster than morphine does. So fat soluble molecules, we can see from this diagram, pass through the blood-brain barrier. We can see fat soluble molecules pass easily, while amino acids and glucose need active transport, right? So if you have a drug that is more fat soluble, then it's going to go faster into your brain. Now, once heroin is in the brain, it turns into morphine. So the actual active metabolite of heroin, for the most part, is morphine. But even though once they're in the brain, they're doing the same thing, heroin has a stronger subjective effect because of the fact that it gets into the brain faster. And whenever you're having a chemical get into the brain faster, that's generally going to increase its subjective effects. So let's see what we say here. Both molecules can permeate the blood-brain barrier, but heroin is more fat soluble, so it passes through the blood-brain barrier more rapidly than morphine does, which gives it a stronger potency. Okay, there you go. So Imodium, some of you may be familiar with Imodium. This is a anti-diarrheal drug that you get at the pharmacy. I mean, you can get this without a prescription, but it's actually an opioid receptor agonist. Okay, so I know that I haven't defined agonist yet. I will later, but basically it's a drug that works on your opioid receptors, okay? And one major side effect of opioid activity is it halts gut motility, okay? So it decreases your ability to poop. So it is people that are, addicted to opiates, well, a lot of times they won't have poop for a very long time, because it causes constipation. Now, obviously, that's not good in normal situations, but if you have diarrhea and you need to be constipated, you need to decrease your pooping, then you'll get something like Imodium. And this is just absolutely fascinating to me, that Imodium is actually working on opioid receptors that are found in your gut, reducing gut motility, but it can't pass the blood-brain barrier because of its particular molecular characteristics. So since it can't get into the blood-brain barrier, since it can't get into your brain, you can't get high off of it, right? So that's why it's not a regulated prescribed drug, even though it has the same mechanism of action, which is activating opioid receptors as heroin and morphine. Okay, so this is amazing. So for sure, on the exam, you are going to need to know that the main purpose of the blood-brain barrier is to protect the brain from harmful chemicals and things like that. And depending on how drugs may be chemically structured will depend on whether or not they're what's called psychoactive or not, if they can get into the brain or not. So heroin and morphine obviously are both psychoactive. Imodium is not psychoactive, okay? So Imodium doesn't get into the central nervous system, but it still does affect your peripheral nervous system in the gut. So, or in your bowels, more specifically. Okay, so again, here is our little concepts and questions that can appear on the exam. you need to know what is Golgi known for. He's known for producing the Golgi stain. Ramon y Cajal, he is known as the father of neuroscience. He is the guy that, discovered that neurons are not contiguous. Their main scientific disagreement was, in fact, that they're, not contiguous. So go through all of these bullet points and make sure that you can elaborate on them a bit. So what do ribosomes do? You don't have to have, again, any information that I didn't talk about, or if it is the case that there's something that you can prove that I didn't talk about and it's on the exam, then that would be a question that would get thrown out, for instance. But I think I'm normally pretty good at showing, just asking questions that are specifically talked about in these lectures. So know all the major physiological characteristics of a neuron. Yeah. So I think it's all here that you need to know. This was a heavier lecture than the first one, and the next couple lectures are going to be like this. And that's because we're talking about the physiology of neurons and the physiology of neuronal communication, and that is kind of complicated, right? So the good news is that this is going to be the heaviest module for the class. So once we're through all of this kind of hardcore physiology and anatomy, then things are going to get a bit easier. I mean, it's still not going to be as easy as a Psych 101 course, but just know that this is kind of the roughest part of the course. So best of luck, and I will see you in the next lecture.